Hello, everyone. Good evening, Soumya. Hi, hello. Good evening, Hita, Trishila, all other people who are in the meet. Good evening, Anuradha. Hello. Hello, Sachin. Hi. So, how are you guys? Are you doing good? Yes, Sachin, nice to see you again and all other people. Okay, guys. Yes, good evening, everyone. So, let me start recording. Hi, everyone. As you know, my name is Ghazala Firdaus, and from now on, I'm the biology teacher responsible for you guys. There have been some shuffling or shifting of teachers. So the previous teachers, Vanna ma'am, which was supposed to take you class, is shifted to another, and I'm here for you guys. Okay. So yes, already we have started the sexual reproduction flowering plants, right? So we will study all the entire chapter and with revision of all the previous chapters that were completed. Okay. So before starting anything, let me just take the technical check. Okay. Just let me know if you're able to hear me properly and is the screen visible to you or not. Okay. So as soon as we take the technical check, let's move on to the class. Good evening, Ashwin. Nice to see you again. Good evening, Likit, Sahana, all other people. Okay, so I guess there is no problem with internet or technical issues. Okay, that's very good. Then let's start today's class. Okay, so without any further ado, yes. Okay, so I think fertilization, up to fertilization, the syllabus is completed. So we will deal after that, that is post-fertilization events. Okay. So before starting the post-fertilization event, I want to have the clarity of how much topics you have understood about the previous concepts which were already done. Okay, so for that, I'll be asking few questions and we will revise about the last class in 1 or 2 slides and then we will move on to the post-fertilization events. Is that clear? Okay, then let's start the questions. Okay, let's start with the questions. So the first question on the screen is like, in a fertilized ovule, N, 2N, and 3N conditions occur respectively. In A option, antipodal, egg and endosperm. B, egg, nucellus, endosperm. C, endosperm, nucellus, and egg. D, antipodals, synergids, and integraments. Okay, so here actually what they're asking is the ploidy of the structure. Okay, they have given few names present in the ovule, structure of the ovule. ठीक है, so उसका तुमको ploidy बोलना है बेटा. So ploidy of the given names. Okay, so the first will be haploid and the second will be diploid and the third will be triploid. Okay, so in this direction, N, 2N and 3N. Okay, so if you have completely studied the question, I will launch the poll then. See, here, first should be N, second should be 2N, and the third should be 3N. I will launch the poll for this question, guys. no need to hurry. Take some time, okay, and look at the options and understand the ploidy. Okay. Don't get confused between egg and zygote, because many people tend to do that. They get confused between egg and zygote. Before fertilization, XL is the female gamete. After fertilization, the XL itself will be converted into zygote after the fusion with one male gamete. है ना? So last 10 seconds. Ashwin, I need you to look at the options once again. Okay, guys, exactly 1 minute is done. I'm ending the poll. Okay, so let me share the results. So results are somewhat like this. Very good, Likhit. You are correct. Okay, 63% people have chosen option B. 32% people have chosen option A. और सिर्फ एक बंदे ने D को choose किया है, C को nobody has chosen. Okay, so let us discuss which is the correct answer for this question. Okay, Anu, you are correct. Okay, guys. So in this question, they're asking about the fertilized ovule. Okay, so ovule before fertilization will be different. After fertilization, there will be some changes in the ploidy. Let me draw a diagram for you. Okay, so all they have given is about the embryo sac, hai na? So I will draw an embryo sac for you here. Okay, so this embryo sac contains 3 antipodal cells. Okay, so these are called as antipodal cells. Okay, and there will be a secondary nucleus, or also called as polar nuclei, or also called as secondary nucleus. Okay, and in the below there will be a bigger XL and 2 sister cells of the XL. This is egg cell, and these 2 are synergids. Okay, so this is the embryo sac. So after embryo sac, what will be there? That means outside the embryo sac, there will be what you can say, a nucellus will be there, a nutritive tissue will be there. Okay, so after nutritive tissue, there will be some integuments, right? So there will be few integuments like this. So यहाँ पे and यहाँ पे integuments रहेंगे. So these integuments okay. So what is this, guys? This is a structure of ovule I have drawn here. Okay. So this is the raphe. So this is raphe. Okay. And this is the structure hilum. Okay. So this structure is hilum. So let it be. What we have seen here, let's discuss about that. And this green color structure is nothing but the protective layers called integuments. So I have drawn a rough structure of the ovule here, okay, along with the embryo sac with very prominent. Let us understand the ploidy number. Okay, they're asking you about the ploidy, guys. See, n दिया है, 2n दिया है, 3n दिया है. Okay, तो यहाँ पे options में आपको choose करना है which is the correct one. Okay, so you can see antipodal cells. So these are the antipodal cells. So let me tell you one thing, guys. This sentence you have to always remember. Embryo sac. Okay, so embryo sac is called as female gametophyte. Gametophyte means what? The structure which carries gamete. Okay, so embryo sac is female gametophyte. Then which is female gamete, ma'am? If you ask me, let me write here egg cell. Okay, so egg cell is called as female gamete. Okay, so embryo sac will become the female gametophyte. That means the structure which carries the female gamete. So female gamete is this egg cell. Okay, So this entire embryo sac female gametophyte, it will contain all the structures that are haploid. Okay, I will write here some another color. Okay, so in the embryo sac, all the structures that you get, okay, so all structures are haploid before fertilization. Okay, so after fertilization, there will be changes. XL will turn into a zygote, that will be there. Now let us come to the ploidy number. Okay, अभी आते हैं ploidy number को. So this is the background that you need to know before answering this question. So antipodal cell. So this antipodal cell is present inside the embryo sac, and I have told you that is a haploid. So antipodal cell is haploid. So egg cell is also haploid. Why? Because egg cell हम जब कहते हैं तब अभी fertilization नहीं हुआ है. Fertilization जब होगा, then this egg cell will turn into zygote. Okay, so endosperm. So when will endosperm come? See, if you have studied about the double fertilization that already you have studied in the last class. So in double fertilization, there is a process called as triple fusion. In triple fusion, what do you see? The fusion of one male gamete with the polar nuclei, which will give the 10 primary endosperm nucleus. And that will be triploid in number. Okay, this polar nuclei is a diploid, right? So this diploid will fuse with one male gamete. So then how many nucleus it will be containing? 3 nucleus, and the ploidy is triploid. Okay, so this one. Now, egg and nucellus. See, nucellus is present outside the embryo sac, right? So outside the embryo sac present है, मतलब ये एक nutritive layer है बेटा. So this nucellus is the nutritive layer which provides the nourishment for the developing embryo sac. Okay, so it provides the nourishment for the developing embryo sac, right? So if it is providing the nutrition, then it has to be diploid in number, है ना? So chromosome number ज्यादा रहेंगे तो क्या होगा? ज्यादा metabolism होगा, ज्यादा protein formation होगा, है ना? ज्यादा amount of nutrients बनेंगे. So nucellus is diploid. Now endosperm is triploid. Okay, so this is triploid. Then endosperm again triploid, nucellus is diploid, egg is haploid. Then antipodal cells, haploid, synergate. Synergate is present inside the embryo sac. That means it will be haploid. Okay, and then integuments. So integuments are present outside the female gametophyte, that is embryo sac, and these integuments have the function of protection. Okay, so these two layers of integuments present in the angiosperm, they will protect the embryo sac. Okay, they will be diploid. Now you tell me which is the correct answer for this question. Okay, so the sequence should be N, 2N, and 3N. Which is the answer, guys? Come on, tell me now. Yes? Lipika, look at the sequence, beta. Yes, Pratiksha, you are correct. So the answer for this question is B. Okay, it is B. Very good. See, egg is N, nucellus is 2N, and 3N is endosperm. Okay, very good, Lipika. Okay, so these are the wrong options. So I hope now you are very much clear about the ploidy. Okay, see, it's very important to understand the number of chromosomes or the set of chromosomes present not just in the gametes but also in the gametophyte. अब देखो, यहाँ पे embryo sac is the gametophyte. Okay, embryo sac is the female gametophyte. Now you tell me which is the male gametophyte and which are the male gametes. Gamete ko chodo, you tell me which is the male gametophyte. Sorry, yeah. Male gametophyte. Very good, Lipika. Pollen grain is the male gametophyte because pollen grain is the structure which carries two male gametes. है ना? So pollen grain के अंदर दो दुल्हे होते हैं. Two bridegrooms will be there. They are ready to get married. So those are two male gametes. Okay, so two male gametes will be there present in the pollen grain. Hence, pollen grain will be the male gametophyte. Very good. Let's move ahead then. Okay, so there is a question on the screen. The total number of nuclei involved in double fertilization in angiosperms are 2, 3, 4, 5. Okay, so I'm launching the poll for this question. Take some time, no need to hurry. I want correct answer. Yes, Ashwin and Anu, what are your answers, beta? Very good, Chiran. But you need to look at the options once again. Look at the options once again. Total number of nuclei involved in entire double fertilization process. They're not asking only about syngamy, they're not asking only about triple fusion. Both the process in entire double fertilization. Very good, Ashwin. I'm happy to see. Okay, so very good. Now Lipika and Monisha changed their answers. Yes, Sita, you are correct. Okay, last 10 seconds. See, that's what I say. You are knowing the answers. Okay, but in a hurry, you choose a different answer. Okay, Lipika, good. Yes, Anuradha, Chiran, correct answer. Yes, Gopika, Anu, Ashwin, all are correct. Okay, beautiful. Right, guys, I'm ending the poll. So let me share the results. So results are somewhat like this. 41% people have chosen option D, 28% people have chosen option B, and 17% people ने C को choose किया है, 14% people ने A को choose किया. See, I hope there is not a good clarity here. Okay, don't worry, we will make the concepts clear now. Okay, so what are they asking, beta? Let's see in the question. They're asking the total number of nuclei involved in double fertilization, right? So in angiosperms, what do you see? You see the double fertilization. Why the process of fertilization in plants is called as double? Because there will be two processes. Okay, so it includes two processes. So the first process is called as syngamy. है ना? So syngamy होता है first. So what happens in syngamy? In syngamy, one male gamete fuses with egg cell to give zygote. है ना? So this zygote is nothing but diploid. Now, so this process of fusion between one male gamete and one egg cell is called as zygote. Okay, then there is another process called as triple fusion. Okay, so triple fusion, it involves one male gamete. Okay, it involves one male gamete with polar nuclei. है ना? So it gives what? It gives PEN, primary endosperm nucleus. Okay, so this primary endosperm nucleus is triploid. Now what happens? So this zygote will give rise to the embryo. As you already know, it will undergo the embryogenesis, and this PEN will give rise to the endosperm. Okay, so this will give rise to the endosperm. Right. Now, so which is the nuclei? You tell me which are the nuclei that are involved here. So one nuclei, two nuclei. Okay, XL from one and one from the male gamete, one from here, and how many from here? So there will be, sorry, 1 plus 1, right? So there will be two nucleus. So 1 plus 1. So now you tell me, so total will be 2 nuclei. So polar nuclei already contains 2, right? So abhi bolo, from here, no, wait, XL will be 1. Okay, so total is 2. Right. So you count me how many nuclei are there. So 1, this one. So total 2 nuclei, and here 1 and 2, total 3 nuclei. So now you add 2 plus 3 is what? 5. So the answer for this question is D, 5. Okay, so all these other options are wrong. If they ask you, okay, how many nuclei are involved in syngamy, you have to write 2 nuclei. If they ask you how many number of nuclei involved in triple fission, you have to write 3. But they're asking about the double fertilization, you have to choose the option D. I didn't understand, Chiran. Ma'am, your video is blocking some part of question. I can't do anything with that. Harsha, next time what I will do, I'll keep the text a little bit lower. Okay? Yes. Chirant, I didn't understand your question, beta. Wait, let me read it. But before that, I'll give the question. Okay, Lipika. So there are some column 1 and column 2 options. So you have to see the terms given in the column 1 and understand the definitions given in the column 2. Okay, so I'm giving 20 seconds for you to read the question. After that, I will launch the poll. Okay, I'm getting answers now. Now I'll launch the poll. There is some network issue. Wait a second. Good, Anu. Now it has launched. Okay. Good, Rushita, Likit, Hita, ... You can add your name better so that we can take your good name. Okay. You had told me that I want to go faster. Okay, I will go faster. Okay. But what I'm doing here is I'm just understanding if you have understood the concept or not. Okay. Because in further topics, I'll be just using the terms. If you don't understand there, then it will be, waste of time. That's why I'm making sure that we have understood everything. Okay. So last 10 seconds. Anu, Gopika, both of you are correct. Yes, Ashwin, you're also correct. Okay, guys, I'm ending the poll. Last 1 second is remaining. Okay, so let me share the results. So 75% people have chosen option B and 20% people have chosen A. And 8 बंदे ने शायद B को choose किया है. Well, not a big deal. Okay, so let's understand which is the correct answer for this question. Okay, so already I had written the structure of ovule in the last slide, right? So there will be a presence of funicle. I'll just roughly draw here. Okay, so integuments will be there. Okay, so after integuments, there will be a nucellus. Okay, so this is the nutritive tissue, nucellus. Okay, so inside the nucellus, there will be a embryo sac. Embryo sac contain antipodal cells, polar nuclei, X cell, and the synergic cells. Okay, so this is the structure of ovule. So this part is called as hilum, guys. Okay, so this is hilum, that is the stalk of the ovule. So the first, sorry, this is funicle. This is funicle. Yes, Lipika. Okay, so hilum is nothing but the point of attachment. Okay, point of attachment between the stalk of the ovule and the body of the ovule. You can say this. Okay, so this part. Okay, so this part is nothing but the hilum. Okay, so hilum is the point of attachment. So let us see here, funicle is the stalk of the ovule. 5 is the option for A. Okay, and yeah, yes. And hilum is the region where both funicle and the body of the ovule gets attached. So it's a point of attachment. So B is for 4. Then integuments. These are the integuments. Okay, so these are the protective layers which protects the embryo sac. Okay, so one or the two layer. Usually in angiosperm, you see bitegmic condition. Okay, right? Take it down if you don't know this term. Okay, so what happens in angiosperms? In angiosperms, usually integuments will be two in layer. Okay, so integuments will be two layered and we call it as bitegmic condition. Okay, and we call it as bitegmic condition. So have you heard about monotegmic condition? Or also called as ategmic. Sometimes there will be no tegma at all. Okay, so bitegmic is usually seen in angiosperms, whereas single tegma or monotegmic is usually seen in the gymnosperms. Okay, and in some of the ovules there will be no tegma at all. That is ategmic, right? So this tegma term, whenever it is, is you are seeing the term that is related with the integuments. Remember that. Okay, so this is C. Now, D is chalaza. Chalaza is actually the part exactly opposite to the antipodals. Okay, or sometimes it is also called as basal part of the ovule. So this is chalaza, guys. Okay, so this part is chalaza. So this is the basal part. Okay, so this is what this part is chalaza. So D here. Okay, the nucellus is the mass of cells which gives the nourishment or the nutrition for the developing embryo sac. So it is giving the food. Okay, so this is E. So answer for this question is A, 5, B, 4, C, 3. Okay, so B is the answer for this question. Okay, so C, D, where it go? So these are all the wrong options. Okay, so these are all the wrong options. Okay, the guys. Now, let's do some revision from the last class because I have to understand how much concepts that you have understood from the last classes. So for the revision, we will understand few things about the ovule. Okay, so we are going to understand about ovule and types of ovule. Once again, just quick revision. Okay, and types of ovules. Okay, so we will see this. And we will see in another part that is, what is that? Types of microspores. Have you heard about the types of microspores? Usually they are not included in your syllabus. Okay, but still we have to study about the microspores. And then we will study about this one. Okay, microgametogenesis. And megagametogenesis. I know you have heard about microsporogenesis and megasporogenesis, but you should also know what is megagametogenesis and microgametogenesis. Okay, so you should know that also. Then, last but not least, we will see some functions of capitum or the speciality of capitum. You will keep answering. Okay, so be active in the chat box. You have to continuously answer for whatever the questions that I'm asking. Okay, so these are the few things that we have to understand. And lastly, the double fertilization, which is already done in your last class. Okay, so double fertilization. Now, right. So look at this diagram, guys. You tell me which is the first diagram here. What is the first diagram representing here? Anyone? Deepika is saying microsporogenesis. Actually, it involves in the microsporogenesis, but it is actually, yes. Okay, so microsporogenesis is what, guys? See, microsporogenesis is actually the formation of microspore. Okay, so microspore formation from PMC, that is pollen mother cell, or microspore mother cell. Okay, so formation of microspore from the PMC or MMC. So microspore will be haploid, but PMC or MMC will be diploid. So this is microsporogenesis. Now, which is given in the first diagram. Now I will tell you what is microgametogenesis. I will write here, guys. So what is microgametogenesis? So what is this? In the term only they have given. Okay, so micro refers to what, guys? See, always in the biology, micro refers to two things. Okay, so usually micro means small, or else micro means male. Okay, gametogenesis, gamete means what? It is nothing but the gamete. Okay, so male gamete. Genesis means synthesis, formation, making. Okay, so formation. So this is microgametogenesis. Now you tell me microsporogenesis. Okay, so I will split the term here for the microsporogenesis. So micro means definitely male. Sporo, or you can say continuously, you can take it as the microspore. Okay, because we are using here microspore. Okay, so microspore is nothing but the immature pollen grain. See guys, now microspore जब भी हम बोलते हैं ना, तो microspore का मतलब क्या होता है? Microspore. Okay, so this is microspore ka genesis. Okay, so genesis means, I told you, right? Genesis means formation. So this is what formation of microspore from pollen mother cell or megaspore mother cell. So this is the formation of male gamete. So how male gamete is happening? See, whatever we get here, microspore tetrad. Okay, so this microspore tetrad or individual microspore, I will write here a microspore. Okay, so this is a microspore. You can call it as microspore, or also you can say immature pollen grain. Okay, so immature pollen grain. So this immature pollen grain has to get mature. So how it will happen? Let me write here. Okay, so microspore will undergo mitosis. Okay, so mitosis division hota hai. Mitosis division hone ke baad, after first mitosis, what do you get? You get one vegetative cell and two generative cell. मतलब एक generative cell आता है. Okay, so this is vegetative cell and this is generative cell. Then after this, there will be a second mitosis. Okay, so there will be again a mitosis 2. You can say mitosis 2. So with this mitosis 2, what do we get? We get two male gametes. Okay, so we get two male gametes from the generative cell. See, name only indicates. Okay, generative cell. Generative cell is going to give the two male gametes and there will be one vegetative cell. Jeevan is saying meiosis. See, beta, meiosis has already occurred. Where? Meiosis has already occurred in microsporogenesis. Ilnodi. Look, Jeevan, here this microsporocyte is nothing but microspore mother cell or pollen mother cell. Okay, so this pollen mother cell will undergo meiosis 1 and meiosis 2. That is two stages of meiosis. After that, what you get? Haploid nuclei. So first, in microsporogenesis, meiosis division will take place. After the formation of microspore, then we get the mature pollen grain. Okay, with two male gametes. So this is called as mature pollen grain, guys. Okay, so this is mature pollen grain. Okay, so this is what? This contain two male gametes. This red color में जो मैंने लिखा है ना, so what I have written in red color, this is two male gametes. Okay, so this is the mature pollen grain. Okay, So I hope, yes, Jeevan, that's why I cleared the doubt. Okay, so from micro gametogenesis, what happened? Mitosis will takes place. So now what is ready? Our two male gametes are ready, मतलब हमारे दो दुल्हे शादी होने के लिए तैयार हैं. So this is about microsporogenesis and microgametogenesis. See, microgametogenesis is also called as maturation of pollen grain. Why? From immature pollen grain, a mature pollen grain is obtaining. Okay, so either two cell stage or three cell stage. So three cell stage is actually called as mature pollen grain. Why? Because now it is ready to get married, है ना? So ये होता है. Now let us understand this one. Now you tell me what is the diagram given here. ये जो नीचे का diagram है ना, ये बोलो बेटा मुझे. So what is this? Either this is the megasporogenesis or megagametogenesis. Which is this? Now you can relate. Tell me, guys, the structure which is given here in the below, which is this? Megasporogenesis or megagametogenesis? I want answer. Look, from one functional megaspore, we are getting an embryo sac. Okay, nobody's saying anything. Well, not an issue. Very good, very good, very good, Sachin, Shalini, and Monisha, Somya, everyone. See, let me write here something. Let me write here. Okay, so megasporogenesis is what? Megasporogenesis. So what is megasporogenesis? You guys tell me. The process of megasporogenesis. Okay, so megasporogenesis is nothing but the process of formation of megaspore from megaspore mother cell. Okay, तो हमें यहाँ पे क्या मिल रहा है? A functional megaspore, right? So we are getting here a functional one megaspore from what? Megaspore mother cell, that is MMC. So MMC is diploid, our megaspore is haploid. ठीक है, सेम थिंग बेटा, जैसा माइक्रोस्पोरोजेनेसिस में देखा वही चीज मेगास्पोरोजेनेसिस में. Now from this functional megaspore, so we got here functional megaspore, right? So this is our functional megaspore. So this functional megaspore will in turn give rise to the mitotic divisions, just as in the microgametogenesis beta. ठीक है, तो अरे what did I get here? Okay, so this, look here. So first mitotic division, second mitotic division, and three. So in megagametogenesis, there will be three mitosis division will occur. Okay, so there will be three mitosis. But in the case of here, there will be two mitosis. Okay, here there will be two mitosis. Where? In the microgametogenesis. Okay, you can see here. So this is here. Okay, so two mitosis here, three mitosis divisions. So after, from 1 we get 2, from 2 we get 4, from 4 we get 8. Okay, so हमको 8 cell यहाँ पे मिल गए. Okay, so in these 8 cells there is some organization. So organization is happening between whom? Between the synergid cells, antipodal cells, and the polar nuclei, and there is also formation of vacuole. ठीक है, so vacuole से formation हो गया. So this is the embryo sac. Okay, so this is embryo sac. So embryo sac के अंदर क्या है? So all the things that we have studied. Okay, so there are two synergids and there is one egg cell also called as oosphere. So this is called as definitive nucleus, also called as secondary nucleus, also called as polar nuclei. Okay, and these are antipodal cells. Okay, so this embryo sac is actually called as polygonum type of embryo sac. Actually, there are many types of embryo sac. Okay, so but in your syllabus they have only given polygonum type of embryo sac. Okay, right. So this is about the concepts that we have understood. Now I told you there are some types of microspores, right? So क्या है types of microspores? See, there are few types. One is tetrahedral type of microspore. Okay, so you can see here 3 microspores. There is present one in the behind. Okay, like it is shown in the 2-dimensional picture, but there is a 3-dimension में अगर हम देखेंगे ना तो पीछे एक रहेगा microspore. Okay, यहाँ पे and here also isobilateral. This is decussate. There will be behind one, this one. Okay, and this is T-shaped one, and this is linear. Usually when we explain the microsporogenesis or megasporogenesis, we show the linear type. है ना? So these are all the microspore tetrads. Okay, so what do you see here? This is nothing but called as microspore tetrad. Okay, so microspore tetrad. So microspore tetrad, see, what you get is a tetrad where microspores are attached to each other. They are not free. So how this microspore tetrad will become individual microspore? Do you know any idea? Do you have any idea? How? There is a presence of a biochemical content here. Okay, that is degraded by an enzyme. Then we get the microspore ke individual cells. Any idea, guys? See, callose, very good. Callose is not the enzyme, Sachin. Callase is enzyme. Callose is the biochemical component present here. See, in all these microspores, there is some structure. Okay, यहाँ पे carbohydrate होता है. Callose मतलब that is one carbohydrate. So that will be present here. Okay, तो यहाँ पे it will make the microspore join together to form a tetrad. Okay, so that is a callose. It's a biochemical compound, callose. So this microspore tetrad has callose, and this callose will be degraded by an enzyme action, the enzyme called as callase. So after the addition of callase enzyme, what we get? We get individual microspores. Okay, so we get what? We get like this, individual and free microspores. Okay, we get free microspores. Microspore. So now all these microspores, okay, so all microspore is called as individual pollen grain. Okay, all microscope is nothing but the pollen grain. So these pollen grains all are fertile. That means क्या होता है, जो माइक्रोस्पोर मेच्योर होके पोलन ग्रेन बन जाता है, मेच्योर पोलन ग्रेन, अभी एक पोलन ग्रेन के अंदर 2 मेल गैमेट हैं, ठीक है, तो उसका मतलब क्या है? That means each one pollen grain will make again 2 male gametes like this. So all the microspores are fertile. But in the case of female, as you see in the female, there was a megaspore tetrad. है ना? Megaspore tetrad में only one will be functional, 3 will degenerate. है ना? I hope you are getting the concept of what I'm explaining here. See here, you are seeing the functional megaspore. Okay, only one megaspore will be functional, and all the 3 will degenerate. Okay, so these are degenerating. That means only one functional megaspore will become the embryo sac. Okay, so remember this one. Here all the microspore are fertile, whereas in megaspore, I'll write here, only one will be functional megaspore. Other 3 will degenerate. Okay, so functional megaspore. So these kind of questions you will get in NEET. Okay, so like I'll ask you a question. Okay, so in what is that, microsporogenesis, you are obtaining 25, like, okay, you have 25 microspores. Okay, you have 25 microspores. So how many male gametes you get from these 25 microspores? This is the question. You have 25 microspores, beta. Okay, so this each individual will make 2, right? One, another one, 2 male gamete, 1 and 1. So 1 and 1, 1, 1. So these kind of questions will be asked in NEET. Okay, so this is your homework for tomorrow. Sit, aram se study the concepts, and then You tell me. Okay, so only Sachin PR has tried to answer. You guys are correct actually. 50, right? So if there are 25 microspores, you are adding one another. Okay, so 1 plus 1 hota hai. So you can multiply with into 2. Okay, you get the 2. That is 50. So for this question, how many pollen grains do you get? Sorry, how many male gametes do you get? For 25 microspores, you get 50 pollen grains. See, this kind of questions you expect in NEET pollen grains. Okay, so I hope I'm clear here. Now, so there are type of ovules. What are the types of ovules? So usually there are 5 to 6 types. मतलब 5 types होते हैं. Okay, so among these types, this anatropous ovule, okay, so this anatropous ovule is also called as inverted ovule. It is also called as inverted ovule. Okay, so this is the most common and it is found in like 64% of the angiospermic families. Okay, it's the most common one. That's why in your NCERT and in your textbooks you study this only. You study this inverted ovule. Okay, so there's another type of ovule called as orthotropous ovule. This is also very common, मतलब less common compared to the anatropous. ठीक है, अगर ये 64% आपको angiosperm families में मिलता है, so you will see this up to some 20%, nearly 15 to 20. We can't say. Okay, that depends on species to species, and these are very rare ones. Okay, these are rare. Okay, so rare, and these are very specific to the specific species and the families. Okay, but the most common is the anatropous ovule. See, I added this structure because in the last class, some of the students asked me, ma'am, what is anatropous? See, anatropous is nothing but the name, type of the ovule. Okay, orthotropous is another type of ovule. In orthotropous, chalaza is towards the earth and micropyle is towards the sky. So here you can see micropyle is towards the earth and chalaza is towards the sky. So orthotropous is just an erect ovule. Anatropous ovule is an inverted ovule. Okay. So what you need to remember is only about orthotropous and anatropous. Okay. So let's move ahead. Now I hope you have already understood the structure of what is that? Megasporangium. Okay. So देखो बेटा, this is the funicle. Okay, so this is funiculus, also called as funicle. I told you that is the stalk of the ovule, and this is the hilum. This entire region is hilum. Okay, so this is hilum, and there is called as raphe. Raphe is nothing but the supporting system, or it will give some mechanical support to the ovule. Okay, so this inner dark color structure that is written, this is just a mass of cells which give the mechanical support to the ovule to stand like this for the erectness. Okay, so this is raphe, and this is chalaza. Chalaza is always present, near the antipodal cell. एल्ली एंटीपोडल सेल रहते हैं अल्ली चलाजा, मतलब कहाँ पे एंटीपोडल सेल है वहां पे चलाजा होता है, और जहां पे माइक्रोपाइल होता है वहां पे एक्सेल होता है. ओके, सो फ्यूनिकल, हाइलम, then chalaza, micropyle, and the nucellus. So this nucellus is the nutritive cells which give the nutrition, and these are the integuments. This is the antipodal cell, this is the central cell, and this is the embryo sac, that is female gametophyte, and these are two polar nuclei. Okay, so I hope you're clear with the structure of megasporangium. Now let me give some understanding about this. So you tell me, which are the layers that you see in the microsporangium, or you can say in an TS of anther. So this is a TS of anther. So what are the layers, wall layers or the anther layers do you see here? Yes, guys? I want people to answer. So first there will be epidermis. So first layer is called as epidermis. After epidermis, what do you get? Endothecium. Okay, so after that you get an layer beneath the epidermis is endothecium. So below the endothecium, what do you get? Below the endothecium. Very good, everyone. People are answering in the chat box. I'm very happy to see that. Okay, there will be either one or two layer. Okay, so these are nothing but the middle layers. Okay, so these are middle layers. Okay, so inside the middle layer, yes, Rishita, now you have answered it correctly. Okay, so inside that we get the this. Okay, so it will contain the tapetum cells. Okay, so this is a pollen sac. Okay, so these are the tapetum cells, that is a tapetum layer, and this entire structure is called as, let me change the ink. Okay, so this entire structure is called as pollen sac, or also called as microsporangium, or also called as microsporangium. Okay, so what do you find inside the microsporangium, guys? Tell me in the chat box. So inside the microsporangium, you will find few microspore mother cells. Okay, so what are these? I will write here. So these are microspore mother cell, or also called as pollen mother cell. Usually there is a tissue called as sporogenous tissue. Sporogenous tissue. देखो पोलन सैक के अंदर जो भी खजाना है ना बेटा, उस खजाने को हम कहते हैं स्पोरोजिनस टिश्यू. स्पोरोजिनस टिश्यू के अंदर सेल्स हैं. वो कौन से सेल्स हैं? Those are nothing but the mother cells. Okay, so these mother cells are diploid. So from these mother cells, what do you get? You get the microspore tetrad, and from microspore tetrad you get the mature pollen grains. Okay, so you get the microspore tetrad. Okay, so from this microspore tetrad, what? Sorry guys, एक second. I will use the eraser. Okay. So from microspore tetrad, what do you get? You get the mature pollen grain. Okay, which contains two male gametes, है ना? So which contains two male gametes. Okay, so now you have understood about the structure of microsporangium and megasporangium. Okay, so बस यही कहानी है. Let's move ahead. Now let us briefly understand about the double fertilization. You guys should be active, so you have to tell me. Okay, so as you know, in pollen grain we have our two male gametes ready. So okay, so here our one male gamete and another male gamete. So our two male gametes are ready to get married. And which are the another opposite side ka bride? Our bride is, okay, so there will be one egg cell and polar nuclei. Okay, so polar nuclei and egg cell are the two female side of people. Okay, ये female side के लोग हैं. Okay, तो ये सब ready हैं अभी शादी करने के लिए, है ना? So they are ready to get married now. So now what happens? So this is the two male gametes are ready, and excellent polar nuclei, everyone is mature and they're ready for marriage. Now, pollination process takes place, right? So pollination is what? The process of transfer of male gamete, है ना? So male gamete से जो process of transfer of pollen grain. Okay, so pollen grain from the anther to the stigma of the gynoecium. Okay, so this is the gynoecium and this part is the stigma. Okay, so our pollen grain after pollination process is sitting on the stigma. Okay, always remember one thing, guys. Okay, so 60% of pollen grains, 60% of pollen grains will be dehisced out. Okay, will be dehisced out of the anther in two-celled stage. Okay, in two-celled stage, 60% of the pollen grains will be dehisced. Okay, so remaining 40% of pollen grains will dehisce in three-celled stage. Will dehisce in three-celled stage. उसका मतलब है, see, pollen grain is ready to release out or liberate out or dehisce out of the anther. So usually that happens in two-cell stage, in the 60% of the angiospermic pollen grains. Okay, so some will dehisce at two-cell stage and some will dehisce at three-cell stage. अभी imagine, अभी हमारा जो pollen grain जो जाके stigma के ऊपर बैठा है, it is a two-cell stage. So now it has to make the pollen tube, right? Ma'am, what are you saying that in 60%? 60% pollen grains in angiosperms. In angiosperms, 60% of angiospermic pollen grains will dehisce in two-cell stage. Okay, Sinchana? ठीक है, तो अब two-cell stages से जो dehisce हुआ है, वो three-cell stage में कब जाके convert होगा? When it is sitting on the stigma. When it is in the stigma, there the generative cell will divide to give the two male gametes. So that division is happening on the stigma. Okay, so this is how, because see, after all, three-cell stage is the mature pollen grain. It has the two male gametes. So for fertilization process to take place, we need two male gametes, right? So male gametes कब ready होंगे? जब pollen grain is in the three-cell stage. Are you clear with this? Yes, everyone? See, I'm not explaining this for the first time, right? You have already studied this in the last class. Now, so yes. So after pollen grain, madam, are you saying that 60% of angiosperms or 60% of pollen grain in one plant? Beta, 60% of pollen grains in angiosperms. I'm not talking about one plant, Sinchana. I'm talking about the entire angiospermic group. There are many angiospermic plants in the environment. 60% of the angiospermic plants में क्या होता है? Okay, so two cell stage में anther से pollen grain dehisce out होता है. Okay, so I'm talking related with the angiosperm beta. Sinchana, is that okay? Now, see, what I'm saying is most of the, okay, thank you. Right, so this pollen grain is on the stigma. Now pollen grain has to produce the pollen tube. So now there will be a presence of compatible protein. So compatible protein will take the part. Okay, so compatible protein present on the pollen grain, it will be act as the identity for the stigma. So stigma will recognize, oh, this is my species of pollen grain only. Okay, so now I will allow this pollen grain to make the pollen tube. See, always remember, when pollen grain sits on the stigma, there will be some interaction between the pollen grain and the stigma. So that is called as pollen pistil interaction. Okay, उसको हम कहते हैं pollen pistil interaction. Right, guys? So how that interaction will take place? See, pollen grain, I will write here one pollen grain just for your reference. Okay, so this is nothing but your pollen grain. So this is XII and this inner one is entine. Okay, so above the pollen grain, there will be the presence of compatible protein. है ना, XII के ऊपर compatible protein रहता है. So ये protein stigma recognize करता है. Stigma will recognize the compatible protein present on the pollen grain, and then it will allow, the stigma will allow the pollen grain to make the pollen tube. See, जो कोई भी pollen grain environment में होता है ना, so वो आके stigma के ऊपर गिर सकता है, but हर हर pollen grain को stigma will not allow to make the pollen grain. Stigma will allow only those pollen grains which the stigma will feel that that is of the same species and that will do something good in the upcoming progeny. ठीक है? Are you getting my point or not? You have heard about compatible protein, right? In pollen pistil interactions. Say yes or no. If you're not responding anything, how would I come to know? Okay. Okay. So our stigma will recognize the compatible protein, and that compatible protein is like the identity card for the pollen grain that yes, I am of your same species. Then the pollen grain will make the pollen tube inside the style. Okay, so it will make the pollen tube and it will reach the ovule, है ना? So it will reach the ovule. So now inside this pollen tube we have two sperms, or also called as two male gametes. We have two male gametes. Okay, so you can see here. Okay, so first male gamete. Okay, so I will write here. So first male gamete is fusing with the egg cell. Okay, so first gamete with egg cell. You can see here. So first male gamete with the egg cell, and that will make the zygote. So you know this. Okay, so this process is called a syngamy. Now you tell me, how exactly is this going to happen? So what is the function of synergid? Can anyone explain? Okay. So can anyone explain what is the function of synergid? Okay. Synergid का function ये होता है बेटा. Let me draw a structure of synergid for you. Okay, so ये एक structure है. This is the nucleus. Okay, so this is the nucleus. Yes, very good. Pratiksha and Deepika, they help in the guiding the pollen tube, and from pollen tube into the entry of the pollen. What is that? Male gamete. Okay, so this is the structure of synergid. Okay, so synergid has this nucleus. This is the nucleus. And there is a filiform apparatus. Okay, एक filiform apparatus रहता है. So this is a finger-like structure. So wherever in biology, if you come across a filiform, that means finger-like. Okay, so filiform apparatus. So now what happens? See, this, let me go to the next slide बेटा. यहाँ पे अच्छा diagram दिया है. ठीक है, तो यहाँ पे क्या हो रहा है? See, so now this pollen tube is ready and it is near the micropyle. Okay, so near the micropyle, synergid cell will make the guidance for the pollen tube. अभी pollen tube के अंदर दो male gamete are there. So that one male gamete, let me write here, one male gamete will enter through synergid with the help of filiform apparatus. Male gamete first will enter through synergids. Okay, synergid के through enter करता है by a structure called as filiform apparatus. Filiform apparatus से वो structure से अंदर जाता है. Okay, so from synergid then it will move on to the egg cell. Okay, so this is the path of entry. So this is how entry will take place. Ma'am, who will guide to the pollen tube? Synergid or filiform apparatus? Monisha, synergid के अंदर filiform apparatus है. If you say filiform apparatus, not wrong. If you say synergid, then also not wrong, because filiform apparatus is present inside the synergid only. Filiform apparatus is just like the door. Okay, so synergid is the one which is attracting the male gamete into the inside of synergid, but filiform apparatus is like the door. From the door, male gamete will enter. Okay, yes. So this is how happen, right? So now you can see through synergid. Okay, so through synergid, the male gamete will go to the egg cell. Let me write here. These are antipodal cells. This is egg cell and this is synergid. Okay, so here we have two male gametes. Okay, so this male gamete will enter here. Okay, through here, and then it will pass on to here. So now syngamy has occurred. Now what happened? This red color will fuses with the green color. Okay, so now this is the syngamy. That means the fusion. Okay, so now is what is happening. ये हो गया आपका syngamy. Okay, the fusion between one male gamete and the another egg cell. Okay, so that will make the diploid zygote. Okay, so XL is turned into zygote. This is one. Now another. Abhi aur ek hai na beta, there is another male gamete. Okay, so this male gamete. Okay, so this male gamete will also again go through the synergate. Okay, either same or either the different one. Okay, that's not the issue. That depends on species to species. So from here also spiriform apparatus is there near the synergate. It will move to the polar nuclei. Abhi kya hoga yahan pe? So okay, so this polar nuclei contain two nucleus and one male. Okay, so this entire structure is called as pen. Okay, so pen is what? Pen is nothing but primary endosperm nucleus. Okay, primary endosperm nucleus. इसको कहते हैं primary endosperm nucleus. So this primary endosperm nucleus is triploid. Okay, so there will be a two polar nuclei. That means one polar nuclei with two nucleus and another from the male. So now there will be a 3N condition. Okay, so this formation of 3N, okay, so this formation of primary nucleus is done by a process called as triple fusion. अब तक तो उसी के बारे में पढ़ रहे थे ना बेटा. So this is formed by the triple fusion. Okay, so that's all. So now did you understand about the complete process of double fertilization? Do you have any doubts? Do you have any doubts? Yes or no? Okay, okay, very good. Now tell me one thing. Have you heard about this term called as pollen kit material? Have you heard this term before? Pollen kit material. Okay, what is pollen kit material then? Okay, so let me do one thing. यहाँ पे मैं इसको erase कर देती हूँ because you have already understood this. So let me write here about the pollen kit material. Is it used for emasculation? No. Emasculation is a different thing, Likit. Okay, emasculation is helped in the hybridization process. Okay, वो दूसरा है. That is removal of anther. Okay, but pollen kit material, I will tell you something about this. Okay, so first point about pollen kit is you are going to find the pollen kit on the surface of the pollen grain, that is exactly on the XI. Okay, कहाँ पे मिलता है आपको? On the XI. Okay, so this is a sticky layer. Write it down, guys. Okay, this is the sticky layer. Okay, found on XI. Found on XI. Okay, so what is the another point? So this is made up of carotenoid and lipid. Okay, so I will tell you carotenoid and lipid. So if you ask me, ma'am, does this pollen kit material is present on all the pollen grain ka XI ke upar? No, this pollen grain material is only, okay, only found in flowers with, इसको block letters में लिखते हैं, with entomophily pollination. That means pollination with the help of insects. I just now noticed that my video has turned off. Okay. Where is my video? My video is not visible on the screen. Well, somewhere it is here and there, I guess. Okay, right. So you can see here, pollen kit material is nothing but the sticky layer found on the XI. See, I had already told you. Okay, so ऐसा होता है कुछ structure. तो एक्साइन के ऊपर क्या होता है बेटा? एक्साइन के ऊपर देयर विल बी कंपैटिबल प्रोटीन. So compatible protein is present in all the pollen grains because it is the label of recognition of that pollen grain. Okay, so compatible protein is present in all the pollen grains, but this pollen kit material is present only in the flowers where the insect pollination is going to take place. Okay, see, for pollinators like insects, color of the flower, nectar, odor, fragrance are the agents. मतलब वो एक attracting करने का चीजें हैं. Okay, so by these things the insects get attracted for the pollination, but also to that this pollen kit material is also present on the exine. मतलब कहाँ पे होता है मैं आपको दिखाती हूँ. So along with the compatible protein, there is also presence of sticky content. Okay, sticky content found on the exine. So when, because of the attraction of these pollen kit, the insect will come and sit on anther. So जब anther के ऊपर आके बैठेगा, because of this attraction sticky material, the pollen grains will be attached to the leg of the insect, and then by that pollination is taking place. So this pollen kit material is very, very important with respect to NEET. They will ask you, where do you find the pollen kit material? You have to write, it is found in the flowers where entomophily pollination takes place. And it helps in the pollination. It is made up of carotenoids and lipids. It is just a sticky layer found on the exine. Okay, so this is about the pollen kit material. So who produces the pollen kit material? Now there comes the functions of tapetum. Okay, I will just take the sheet here because I have not taken the sheet. Okay, so I will write here. Now you guys tell me what are the functions, guys. One by one, very quick. Functions of tapetum. First one is it gives nourishment. It gives nourishment for developing pollen grains. Come on, write in the chat box. Yes. It will produce. Very good, Harsha. Ubish bodies called as sporopollenin. Very good. Secretes enzyme called as callase, which works on the callose. Okay, callose के time पे उसको degrade करता है. Then let me change the color again. Okay, so what else? It produces the hormone. It produces a hormone called auxin. Okay, so this auxin is helpful in the growth and development of pollen grain. Okay, so this is another function of the tapetum. So what else, guys? So another thing is it provides, okay, tapetum provides this one, compatible protein that is found on the exine. Okay, so compatible protein that is found on the pollen grain. Last but not least, what is this? So it also gives or makes the pollen kit material. Let me change the ink. Okay, so it will gives the pollen kit material for the insect pollination plants. Okay, pollen kit material is produced. So what is the function about, what is the speciality of tapetum? Yeah, it somehow helps in the pollen maturation. Pollen maturation में help ऐसे ही करता है बेटा. तो compatible protein बनाता है. Okay, भाग्य, it will make the compatible protein. It will release the hormone. It will release the enzyme. It will give the pollen kit material. Okay, so there is something called a syncytial condition. This is very, very important, guys. What is syncytial condition that is found on the tapetum cells? Yes, anyone? Syncytial condition is called when there is a presence of more than one nuclei. Okay, so I will write here syncytial condition. Okay, so presence of more than one nuclei, presence of more than one nuclei. So as you know, okay, usually tapetum cell is a binucleated condition. Okay, so I will write a cell here. Okay, so this is the tapetum cell and it will be like this. ठीक है, ऐसे circulate करते हैं. Tapetum cells will be like this. Okay, so syncytial condition is the term used for the, used where there is the presence of more than one nuclei. Okay, so usually a tapetal cell is a binucleated condition. Well, in some other cells, okay, in some other species, there will be a multinucleate condition is also present. Okay, so this is about the tapetum. Now, I think if you have clear understanding about all these things, then now we are ready to enter into the post-fertilization event. Okay, right. So now, very good. Yeah. So let's understand about the post-fertilization events. So what are the things that you see in post-fertilization events? Yeah, there will be an endosperm development. This endosperm development is happened by a pen. Okay, and embryo development is happened by the zygote. Okay, then so ovule is converted into seed after fertilization, and ovary is called as fruit. So these are all the post-fertilization events. Okay, that we will see one by one now. Right. So you can see here. So these first row. Okay, so this one is called as parts before fertilization. Okay, fertilization से पहले क्या चीजें हैं? Fertilization के बाद क्या है? वो given in the second one. Okay, so here you see petals, sepals, and stamen, stigma, and style. So they will wither off after the fertilization. See, sepal का function क्या है? Sepal is helped in the protection. It will give the protection for the floral bud. Then petals are helped in the attraction of the pollinators with its bright coloring. Stamen is the androecium, which has the, stamen is the unit of androecium, which contains the male gametophyte. And then style and stigma is involved in the female part, that is gynoecium. जो कि ये सब after fertilization के बाद it will wither off or fall off because it is not, it is not used now. Now its work is done, so they will fall off. Now the ovary, see, this is the structure. Okay, so this part is called as ovary. Okay, so this ovary will turn into fruit. Okay, and the presence of ovules. Okay, so what are the ovules present inside the ovary will be turned into seed. Okay, so this I will write here. So ovule will turn into seed. Now, so this is done. Okay, so ovary and ovule. So egg is converted into zygote, that is a diploid condition, very important. Okay, then funicle, the stalk of the ovule is now called as stalk of the seed. है ना? So if you can take the example, I will write a pea plant. मतलब pea, बटानी करके बोलते हैं ना, बटाने pea. Okay, so this is the placenta, marginal placenta. Okay, तो इसका ये छिट्टा होता है. Okay, this is the peel like this. So this is actually, what is this? This is the stalk of the seed, which was initially the stalk of the ovule. ये देखो बेटा. Okay, so what is this? This is nothing but the stalk of seed. Okay, so this batani is the seed. ये जो batane है, क्या कहते हैं उसको? Beans. Okay, so this is the seed which was initially a ovule. So micropyle, ठीक है, जो micropyle था in the ovule, after fertilization it is helpful in the facilitation of oxygen and water uptake. Then nucellus is turned into perisperm. This is very important, guys. See, nucellus is the tissue or the structure present outside the embryo sac. Okay, while there is a development of embryo sac, the entire nucellus is used up. Okay, there is no remaining of nucellus. A condition. Another condition, even after the embryo sac get matures by using the nucellus, फिर भी कुछ amount of nucellus बच जाता है. That will remain. Okay, so perisperm is called as remnant of nucellus. मतलब कभी embryo sperm embryo sac develop हो जाने के बाद भी कुछ amount of nucellus बच जाता है. Okay, so that is called as remnant of nucellus. You have to understand this term very carefully. There will be many terms: perisperm, pericarp, apospore, agamospore, then parthesis. Okay, then what is that? Parthenocarpic. There are many terms, beta, that we will study now in the upcoming classes. Okay, so integuments will turn into seed coat. Okay, so what happened here? Integuments will turn into seed coat. Integuments will turn into seed coat. So the outer integument is called as testa, and the intertegument is, seed coat is called as tegmen. Then synergate cell and antipodal cell will degenerate. I told you, right? Synergate cell का जीवन का उद्देश्य क्या था? Okay, what is the ultimate function of the synergate cell? To help in the guiding the pollen tube and in the entrance of the male gamete. So male gamete enters through the synergate and then reaches the excellent polar nuclei. So after that, they degenerate, and this secondary nucleus will turn into the endosperm, and this endosperm is triploid. So these are the post-fertilization events. All the things that I've explained. See, guys, we are remained with a very less time. So we'll do one thing. We will start this development of endosperm in the next class. Okay, so we have to study about how endosperm is going to develop. Okay, and what are the other I told you, ovule in the fruit, ovule se seed jo hota hai, fir ovary se fruit jo hota hai. So sab changes kaise hote hain, we'll study that in the next class. Okay, so right now there is a question on the screen. So which one of the following events takes place after double fertilization? Read the question carefully. They're saying after double fertilization. Now, options are A, the pollen grain germinates on the stigma. B, the pollen tube enters the embryo sac. C, two male gametes are discharged into the embryo sac. D, the primary endosperm nucleus develops into endosperm. Okay, Monisha, I'm launching the poll now. Monisha, don't worry, beta, I will explain that. Okay, you just right now, abhi iska sawal ka dekho, beta. I will explain that when we look into the structure of the seed, okay, and the fruit. There I will explain you, Monisha. Okay, now I'm just giving you the overview that if any nucellus is remained, even though the embryo sac is completely developed, that remaining of the nucellus is called as perisperm. Now you just know this. How that will look, what are the examples, where do you see the nucellus? Okay, what do you call those seeds? These are called as actually perispermic seeds. वो सब मैं आपको बाद में explain करूंगी. Okay, Monisha, don't worry. Now, okay, last 20 seconds. Very good, Ashwin, Gopika. Anu, I'm expecting answer from you also. Okay, your time is done. I'm ending the poll now. So let me share the results. So results are somewhat like this. 95% people have chosen option D. I'm so happy because initially you were choosing the wrong answers. Now I hope you have got the clear understanding about the double fertilization. Okay, so let me stop sharing this and discuss which is the correct one. Okay, so they're asking after fertilization ka event, matlab double fertilization ke baad wala, okay, takes place after double fertilization. So pollen grain germinate on the stigma. Okay, so this is the process that involves in fertilization, right? So that's why this is not the option. क्योंकि pollen grain जब germinate नहीं होगा, then how does the fertilization takes place? So this part is involved in the double fertilization. Then pollen tube enter into the embryo sac. This is also a part of double fertilization. Then two male gametes are discharged into the embryo sac. This is also the part of double fertilization. Now, when primary endosperm nucleus develops into endosperm, this is actually what? This is the process or event that takes place after double fertilization. मतलब double fertilization के बाद वाला change. जैसे from zygote, from single process, what do you get? you get by the fusion of one male gamete and XL, you get zygote. And from zygote, you get the embryo. Zygote to embryo is after fertilization process, that is post-fertilization. Here also they're asking, from pen you get endosperm. So answer for this question is D. Okay. So I wanted to ask one more thing. Do you have any idea about the layers of XI? Tell yes or no. Layers of XI? No. See, guys, before ending the class, there are still 4 minutes remaining. XI is made up of 3 layers. Okay, so this is important. In most of the books you don't find it, but I hope this is necessary for NEET. Even though this is an extra information, I feel like you should know this. Okay, so the first or the outer layer is called as tactum. Okay, so there is a outer layer called as tactum. Okay, and middle one. Okay, and the middle layer is called as baculate. Okay, and the lower, or you can say the inner. Okay, so the inner layer is called as foot layer. Okay, so this is called as foot layer. Now, these upper 2 layers, okay, so these upper 2 layers of the XI, they are what? They are interrupted by germ pore. Hence they are discontinuous. Okay, अभी देखो बेटा यहाँ पे एक intine है. Okay, so there is a exine. Okay, so exine mein, this is a foot layer. So after this, there is a. Okay, so here you can see this foot layer or the inner layer is continued. Okay, so this is continuous attached with intine. Okay, attached with intine. Why this is continuous? Because it is not disrupted by the germ pore. Abhi dekho, this exine, this is the germ pore, right, guys? This is the germ pore. So germ pore is interrupting these 2 layers. Now, these layers of the exine actually help in the pelinology. Okay, have you heard this term called as pelinology? Pelinology is the branch of botany which involves in the ornamentation. Okay, which involves in shape, structure, and ornamentation. Shape, structure, and designing or ornamentation. There are many types of ornamentation. There are many types of pollen grains, beta. Okay, so there's an entire different study for that. Okay, ornamentation. Okay, so this is about the pelinology. So I thought that I should tell you about this. Okay, so yeah, you can see floriculture. So floriculture is a different part. So it is a branch of floriculture also. So these are all the layers of the exine. Okay, so now I'm launching the teacher NPS. Please make sure to give the feedback before leaving the class. Okay, so if you have any doubts, you can ask me in the chat box meanwhile. Okay. If you have any doubt, please make sure to ask in the chat box, or else you can leave the meeting. Thank you, Anu, Ashwin, and Gopika Vedant for your presence in YouTube. Okay, welcome, Anuradha. If you have any doubts, you can ask me. Yeah, definitely, Jayant. I will share the class notes. Don't worry about it. Okay. So from tomorrow, we will start the post-fertilization events, that is development of endosperm. There are types of endosperms and sorry, all these things. Yes, Likit. Okay, there is a question here. Let me read. It's a much bigger one. Ma'am, hibiscus plant produces bisexual flowers, but it does not produce seeds. And a new hibiscus plant can also be obtained from the vegetative propagation. Then what is the need of producing flowers? What is the function of flowers in this plant? Sinchana, actually, time is done. For this, I need to explain something. Okay, so you people think that hibiscus plant doesn't produce seeds. That is wrong. You go and search today. I will tell you the answer tomorrow for this. Okay, so because I need some time because time is already over. That's why. Okay, I'll get back to you with this tomorrow. Right, Sinchana? Okay, guys, so time is done. Thank you for your presence. I'm ending the poll now. Okay, so we'll see you tomorrow.